Ma'am, shall we start?
Yes, ma. Yes.
Science is a beautiful gift to humanity. It's a famous quote by Dr. APJ Abdul Kalam. A very pleasant morning wishes to everyone connected here virtually. Marian Star Center, the Department of Chemistry, St. Mary's College Autonomous Thoothukudi is happy to organize this third day of the Faculty Development Program on exploring new frontiers in chemical and biological sciences under DBT Star College scheme sponsored by the Department of Biotechnology, Government of India, New Delhi. We welcome you all. God speaks in the silence of heart. Let us invoke our Almighty who has clubbed all of us in this ritual FPP. Prayers.
I think your mic is mute. Madam.
Sorry, sir. If the sign on your heart says welcome, the love will come pouring in everywhere. Now, I call upon Dr. A. Parveen Sultana, Assistant Professor of Chemistry, to welcome all.
In the name of Lord Almighty, I feel this moment a great privilege and honor for me to extend a hearty welcome to you all at the third day of 1 week faculty development program on exploring new frontiers in chemical and biological science, organized by Marian Star Center, Department of Chemistry, St. Mary's College, Autonomous Thoothukudi, sponsored by DBT New Delhi. I extend a very warm welcome to our Secretary, Reverend Sister Flora Mary, our Principal, Reverend Dr. Sister ASJ Lucia Rose, Deputy Principal, Dr. Sister Kulande Teres, Overall Coordinator of DBT Star College Scheme and Member Secretary, Dr. Sister Oroke Genesius Alphonse, Head of the Department, Mrs. S. Rani J. Mary. I especially welcome the faculty members of our college and other colleges who are connected here for FDP. Many of you have made a huge effort to join us today. On behalf of all, we are deeply appreciative and offer you our most grateful welcome to the expert speaker of today's session, Dr. P. Yuvraj, Senior Scientist, CSIR, Northeast Institute of Science and Technology, Branch Laboratory, Impal, Manipur. Sam has done his UG and PG studies at University of Madras, Chennai, MPhil at Vinayaka Mission University, Chennai. He has done his PhD in synthetic organic chemistry at University of Madras, CSIR, CLRI, Chennai, and his postdoctoral fellow at University of KwaZulu-Natal, South Africa, and IIT Madras, Chennai. His areas of research interest are synthetic organic chemistry, medicinal chemistry, asymmetric synthesis, total synthesis of natural products, development of new synthetic methodologies, organic materials, and dyes. With his research experience, he has served as project assistant in CSIR CLRI Department of Chemistry, as scientist in Syngene International Limited and Biocon Bristol Myers Squibb R&D Center, a subsidiary of a Biocon company, as a junior analyst at Thomson Reuters India Services Private Limited. At present, he is working as a senior scientist. He has done many external projects, CSR mission, mode projects, CSIR in-house projects. In addition to his academic records, he has qualified GATE in 2010, CSIR Senior Research Fellowship in 2012-15, postdoctoral fellow from University of KwaZulu-Natal, South Africa, and IIT Madras, Chennai. He has shortlisted for CSIR Young Scientist Award 2019 in Chemical Science. He has published more than 30 papers in the international peer-reviewed journals. In addition, he has presented many papers in conferences and has done poster presentations and has attended many workshops. He has also been invited as resource person in many invited lecture series webinar. We are fortunate enough to have such an eminent resource person today. We welcome you, sir. Once again, I welcome you all. Thank you.
Thank you, ma'am, for your warm words of welcome. Dear participants, kindly switch on your video for the photo session. Thank you all. Now I am delighted to invite the resource person of today's session, Dr. P. Yuvraj, Senior Scientist, CSIR, Northeast Institute of Science and Technology, Branch Laboratory, Manipur, to deliver the lecture on synthesis of biologically active small molecules using sustainable pH activation approach and their medicinal application. Sir, the session is yours, sir.
Yeah. So good morning to all of you. Thank you very much for your nice introduction. And first of all, I thanks to organize committee, so giving me opportunity to present my current research work. So I hope you are getting my slides, right?
Yes, sir. Yes, sir.
Yeah. Okay. Yeah. Today I am going to present my research work, what currently I am doing in my research laboratory. This is one of the piece of my research work who is doing my one of my students. Synthesis of small molecule, the bioactive small molecule that using CH activation and their medicinal applications. Before going to start my presentation, just I want to share what I am doing currently in my laboratory in my research work. This, all are well known about current pandemic situation. As a CSR scientist, just we are working in the current pandemic like this COVID-19. So we are now started to research in already existing the molecule repurposed drug. Just we are working on that. We want to use already existing drug candidates for this current pandemic. So just I want to share some 2 slides about what I am doing currently in the research, like this COVID-19. So we all are know about this is a viral disease. Just we want to use already existing the viral drugs in our globes. Okay, what is the repurposed drug? So how can we use the repurposed drug into the current situation? So how can we modify it? How can we use? So what are the, how can we stabilize the molecule? And these all are the facts. So how can we repurposed drug will work? So we can use some combination of drugs and how to delivering the drug and dosage. Dosage means the mg wise you can increase. Already we are using for suppose 50 mg. This current the current virus COVID-19 we can use 100 mg. Just I am saying the example. We can make dosage. What is the? Just I want to share the repurpose. How to repurpose the drug and formulation. So these all are the drugs currently we are using. Antiviral drugs. All are the Favipiravir, Remdesivir. These all are the drugs for using with the combination of drugs, antiviral drugs, antiparasitics, antidiabetics, and antibiotics. So many combination is there. So we want to use already existing drug molecule with a combination. We want to come up with some good result. So I am going to explain. These are the 12 molecules. Already explained. This is a Remdesivir, Favipiravir, chloroquinone, hydroxychloroquine. These all are the 13 molecules we are chosen for the repurposed drug. So now all the CSR we are having. We all are. I think you are all know about. We are having CSR having some 38 laboratory. In that 9 chemical science laboratories. So in that 9 chemical science laboratory, we are going to synthesize this all the 13 molecule. Just if we want apply for this current pandemic COVID-19 against of COVID-19. So we want to check so how it's work. So already trial is going on throughout the country. So we want to give the tablet formulation. These are the current situation, and currently we are working in my laboratory. In my laboratories, we are working. We have taken one molecule. Just we are working. This is my current field. So just I want to share. I am working in 3 different fields. One is the small molecule synthesis, and also some organic materials like we can use as a OLED. And so many applications is there. That is organic materials, and one is small molecule as a medicinal chemistry. So in this, I am going to today I am going to talk about CH activation. What is CH activation? So what are the application? What are the advantage with the conventional method? First, we want to check what is the metal catalyzed organic reaction using homogeneous catalysts. I think we all know about what is catalyst. Catalyst means it won't react with your compound, right? It will decrease your activation energy, right? We all know about, suppose this is EA, your activation energy. This is your activation energy. This is your product. This is your reactant, right? Suppose if you are using any kind of catalyst, it will decrease your activation energy. It will form the product. This is our idea. This is catalyst. So in this session, I am going to talk about homogeneous catalyst. That catalyst using for CH activation. So as if generally that a chemist expect to synthesize the molecule with efficient manner, right? The reaction should be the highly economical manner, and it should be environmental friendly, the reaction. So what is that means? So you have to use non-toxic solvent and reagent during the synthesis, and there should be a less waste or side product. This is our main motto. So if you're seeing the conventional method, if you want to synthesize the molecule or drug, it will be a multi-step process. We all know that it's a tedious process. So we want to overcome, we want to use less steps. So how can we synthesize the molecule using simple step? Like we need to get that our desired product. It is very difficult to synthesize the natural product or biologically active molecules or chiral molecules are 5 conjugated, the chromophores. However, those molecules are highly useful. There's a many applications. We all know that. There is no direct access for those molecules because of the conventional method. So it's a multi-step, the tedious process. The homogeneous catalyst, they play the greater role. I'm going to explain after that what is a homogeneous catalyst, how it's minimized our steps, right? This first, the first involve the design of catalyst and developing the methodology for the target of compound. This is our motto. You can see this is catalyst design, synthetic methodology, and we want to investigate what is the mechanism. So what is our idea? We want to synthesize the natural product or biologically active heterocyclic compounds, the chiral molecule, or pi conjugated system for using for some kind of what I can say as a fluorescent molecules. So this is the catalyst evaluation. We all know that this is chiral catalysis. This is the, you can see these all are the person or eminent scientists or Nobel laureate working the evaluation of the catalysis. You can see this one, the Williamson catalysis. We all know that Williamson catalysis is a chiral catalysis. He got Nobel Prize in 2001. And after that, cross metathesis is a Nobel Prize chemistry in 2005. This is we all know that Krebs catalyst. And cross coupling catalyst, we all know that we are using Suzuki egg reaction. These all are the catalysis every the decade is developing. So what are the advantages? And finally 2021, you know, we all know that the Benjamin, he got asymmetric organometallic catalyst for Nobel Prize. So these people are inventing different type of catalysis. They want to minimize the process. So we need a speediest process. We want to minimize the steps compared with conventional methods. These all are the just simple introduction about what is the catalysis. Suzuki coupling, we all know that Krebs catalyst or what organic metallic catalysis. Just I'm going to explain what is the CH activation. CH activation is defined as a cleavage of unreactive CH bond and then followed by CC bond or CX, X means N, O, or N. The bond formation obtained a desired functionalized molecule. So as we can see the image, you can see that CH bond is really very difficult to break this bond, because this is sigma bond. We all know that this is required a lot of energy. The CH activation, so we are using that CH activation, the purpose we are using for transition metal catalyst, right? So it's a thermodynamically stable. You can see that benzene or whatever the alkane or alkene, this CH bond is thermodynamically stable, so we cannot break. So if you are using catalyst, that organometallic catalyst, transition metal catalyst, we can weaken the CH bond. So then we can use for next the further process, means that CH activation. This is our main motto. This is the CH activation about general introduction. The advantage, what are the advantage of that CH activation? The CH, the reaction is one step process. If a conventional method, it will take 2 or 3 steps. If you are using the CH activation, it's the one step, the single process. You can see here, I am explaining here. And after that, no need any pre-functionalized starting materials. This is the one advantage. And the important step is we can use the more functionalized molecule. We can make the different substrate, different kind of molecules, different kind of product using that CH activation. These all are the advantage compared with the traditional what we are using the recent reactions. We want to synthesize a different product. These all are the advantage why I am choosing that CH activation. So one more thing, if you want to synthesize as a natural product, so we are going to mimic the natural product. So what are the possible ways is there? Suppose you can choose different reaction, naming reaction. Suppose just simply I explain. If you want, if I need just I explain here. FG. This is different functional group. I need this kind of molecule. Just I am telling. If you want to make any conventional method, just we know that we have to take some benzene, nitrating reagent, BLR H2SO4 nitric acid. We have to make nitro group, then reduction, amine, right? I think I am making clumsy, right? Amine. After that, we have to make Sandmeyer reaction. We can make these different functional groups. We can make here Cl, chlorine, bromine, amine, or whatever the thing we can make the functionality. This is the conventional method. It will take tedious process, right? So if you want to overcome, I need that product without 1, 2, 3, or 4 steps, then I will get the desired product. Just I want to overcome. So what are the reaction is there? So how can I make the desired compound without this stepwise process? That is, so we want to functionalize very easily. That purpose we have chosen the the CH activation, and one more reaction we all know about MCR reaction, multi-compound reaction. So this both we can save that steps, right? If you are using that multi-compound reaction, you can take all the reactant, we can mix in the one product, we can get the different kind of substituent in the single molecule. If you want to synthesize in the same molecule, minimum it will take 2 or 3 or 4 or 5 steps. If you are using the multi-compound reaction, you can get the single steps process, you can get the desired product or whatever the what you are expecting your molecules. These are the advantage. The one more thing, that CH activation. So this is our idea to overcome advantage or disadvantage of conventional method. So we are using the CH activation. So in this CH activation, we can choose our directing group. If just I will explain here. This is R group. Yes. Yes. These all are, we can activate. This is CH activation, right? Suppose if you are using this molecule, it will take, we won't get any selectivity of product, right? Because CH activation nowadays we are working in ortho, and now we are started to working in meta, that it will coordinate with your metal in terms of this ortho position, para and meta. So if you are using a catalyst or palladium or ruthenium or whatever the transition metal catalyst, you can activate the CH bond. Suppose if you are using the CH bond, you won't get the same product, because if you are having three CHs there, right? So we won't get any selectivity product. That purpose we want to make a directing group. Suppose I want one desired product. I want a desired product. That purpose I need where that I want to do CH activation. That purpose I need directing group. So what is directing group? It will be helpful for this reaction. So CH activation is approached due to the unique nature of CH bond in the molecule, right? This is our directing group. This directing group, it will activate here. If you want your ortho substituent or para substituent or meta substituent, we can activate here this ortho CH bond. It will helpful for this the product formation. Suppose if don't have any directing group here, if I am telling here, or if you are having any no directing group, you won't get the desired selectivity. That purpose you need what I can say directing group. The directing group will direct your desired product. So where you want that product. So where you want to do CH activation. So this purpose we are using this directing group. It's important to keep this directing group. It won't attach to your compound. After finishing your reaction, you can remove the directing group. You will get our desired product. This is because the CH activation is the very I can explain. It will take a lot of time. Just because due to time constraint, just I minimize the explanation and all those things. In the CH activation, we need directing group. Directing group will be helpful for where to attach your compound or alkene or alkane. That purpose. And we are using transition metal complex, means palladium, ruthenium, rhodium. So many we are concentrating. Nowadays we are working on like cobalt, manganese, because the rhodium, ruthenium, palladium is a costly catalyst. Just if we want to overcome that, we are changing that transition metal into like cheap metal like iron, cobalt, nickel, or like that. We are working. That I will explain maybe after that. So just I want to explain here is a directing group. It will be a direct to the desired product. So it will form some that metal with chelate with your directing group and metal and CH activation. It will form the like a 5-membered intermediate that finally you will get your desired product. Already explained. This is you can make a different kind of product, means you can change different varieties. So this is the basic principle of CH activation. So you can see the directing group, it will play leading the regio selectivity of CH activation adjacent to your particular directing group. In general, directing group assists the transition metal CH bond activation involve 5-membered is a cyclometal chelated intermediate. So after that, I will explain in that mechanism part. So how it's forming the intermediate. So we all know that CH activation means it's oxidative addition and reductive elimination. That is the process. The two reaction it will take place in the single the process CH activation, right? So after that, so annulation. What is mean by annulation? The CH activation is having some 2 or 3 process in the single step. The directing group or annulation means is a cyclization, nothing but cyclization. After CH activation, we can cyclize the molecule. This is the process called annulation. The transformation involving the fusion of new rings to the molecule via two new bonds. And the type include 2, 3, or 4 carbon atom annulations. The functionation and the ring formation of aromatic and heteroaromatic alkanes are highly important in organic synthesis, right? Those type of methodology are widely used for synthesis of many natural products or biologically active molecules or pharmaceutical application and agrochemicals and organoluminescent materials. Those molecules can easily achieve via this metal catalyst cross coupling reaction. So what are the advantage if you are using CH activation? What are the application we can use for using this CH activation? Because different application. So if you want to synthesize any natural product or medicine or agrochemicals or pharmaceutical intermediates, nowadays is a very booming field. We all know that LED, organic LED, luminescent material, fluorescent material, phosphorescence material. So this purpose, the CH activation is helping more because we can make some more pi conjugated molecules. Like, see, this is the simple mechanism I'm explaining here. This is our molecule. This is you are going to couple with that alkene. It's a metal oxidant, right? You can see this is the intermediate. I will explain the mechanism. Finally, it will form the annulation. Annulation means a cyclization of your molecule. You will get the new ring to attach it to your starting material. In this, the CH activation, we plan to develop a novel methodology for construct of polyheterocyclic molecule using CH activation using rhodium or ruthenium catalyst, incorporating a pi conjugated moiety with a polyheterocyclic core, enhance the emission profiling. We all know that if you are increasing your double bond or pi conjugation, if you want to synthesize any kind of fluorescent molecule, it will enhance your fluorescence activity, right? That purpose, the CH activation is more powerful method for increasing your emission profile. And we can measure the fluorescent property and the AI aggregated induced emission, the property as in this solution state or solid state. Finally, the selected compound can be utilized for the, what is the application? In this, I'm going to discuss here. I'm going to do some kind of live cancer cell imaging studies because my molecule is fluorescence active molecule. This is the scope and development of CH activation, what recently we have started in our research group. So this is, I'm going to synthesize. This is the coumarin moiety, right? Coumarin, we all know that coumarin. Coumarin is a naturally occurring compound. It is different kind of medicinal properties. So based on that, we have chosen this molecule. We want to enhance the pi conjugation or fluorescent activity. So we all know that the coumarin might exhibit the wide range of biological property, right? Already I discussed. So like anti-cancer activity, anti-HIV, antibacterial, antifungal, anti-inflammatory, antioxidant. So many properties there. Based on that, we want to move on, work on that coumarin moiety. We want to enhance, we want to do that CH activation. We want to enhance the emission profiling. That moiety with coumarin and also with emission property. So based on that, just we are just making that molecule, and the that isocoumarine are widely isolated from the, is isolated from the fungi, bacteria, plant, insects. So based on that, so just we are synthesized in our research group. Although this numerous report already exist using this ruthenium catalyst or rhodium or whatever the thing. So only few report available for synthesis of biologically active this isocoumarine. So based on that, we inspired, we want to synthesize the coumarine moiety with the 5 conjugated system. So this is our initial the reaction. So you can see here. Yeah. We are using ruthenium as a catalyst, the synthesis of biological activity, the coumarine, which displays the biological properties such as already explained anti-cancer or anti-HIV. The first we reported influence of ruthenium catalyst in this synthesis. Moreover, you can see here, there is no report on the synthesis of 8-amino isocoumarine, but using the CS activation. We started the preliminary reaction with the isotactic anhydride and diphenyl acetylene, this copper acetate and ruthenium complex, and DCE 110 degree. So we are getting desired product. This is our preliminary reaction setup. So whenever we start the reaction, just we will start, just we want to check the reaction is going or not. No, just if you're doing any literature survey, so we will get if you're doing literature, we can find this is a feasible. We can theoretically we can choose this reaction will go. So we want to do some kind of trial test, trial reaction. Just we will identify first the product. So we started. This is our trial reaction. So we got desired product in 18% 3A. So this is my directing group. Already explained, this is my directing group. So how it will form this coumarine moiety, I will explain the later. So after getting the product, just we want to go for the identification purpose. We want to first identify the compound, the product is formed or not, right? That purpose we went for NMR or C13, mass spectra or HRMS, single crystal XRD, whatever the thing. The preliminary, after set up your reaction, we want identify the product. So you can see this is my product, desired product. We taken C13 and NMR in chloroform. So these all are aromatic proton appear in the aromatic region. This is our these amine compounds. And the C13 molecule, you can see we are having one carbonyl carbon. The carbonyl carbon appear at is 163. Just to be confirm that this is our desired product. And this is our HRMS. This is our exact molecule. This exact mass 313. So we are getting M+1 pattern. So we are getting the 314. So after confirming our product, so we want to optimize the reaction, right? So because I'm getting only for 18% of product, so we want get at least minimum 90% of product. That purpose, I need to optimize the reaction condition. So I can change like catalyst or what I am using, oxidant or solvent or temperature. So I won't get better result. Means I need finally better yield. That purpose I want to do some optimized condition. That purpose we started to some optimum condition. In this, after completing the preliminary reaction, we observed that we got very less yield, right? So we tried optimum reaction, the various conditions, they improved the yield of the reaction. The first we fix the catalyst, right? So first we have to fix the catalyst. So remaining just I want to change oxidant or solvent. So based on that, you can see here I have changed first solvent DC, methanol, toluene, or DMF. So polar to non-polar or high boiling solvent are like that. In this, you can see here that so DMF as the solvent, we are getting 80% of yield, right? So when we use DMF as a solvent, we obtained is a very good yield. Surprisingly, this condition provides around 80% of yield. Therefore, we decided this is our optimum condition of the reaction. So after that, we want to make this substrate scope. What is the substrate scope? We can change different substrate, electron withdrawing group. So I can use here. So this also I can change the phenyl electron withdrawing, electron donating. So what will happen? The yield. So what are the substrate scope? Because if I want to go for any kind of studies, biological activity studies, at least minimum I need some around 20 to 30 compounds. Then only I can compare. Because now I am going for this live cell image studies, right? So I need different substrate. So using this optimum condition, so I want to do different kind of substrate scope. You can see here, I make different derivatives, different substrate. So using different like a substitute in the compound A or 2 is phenylacetylene. So we are changing electron withdrawing group or donating group, aliphatic or aryl. Just we are playing in the wherever it's possible. We want some kind of substrate, right? As we got the optimum condition, the reaction. So we further explore the substrate scope of the reaction by placing the various isotactic anhydride, but keeping the phenylacetylene is intact. You can see in this format. Just we are keeping phenylacetylene is the same. We are changing the isotactic anhydride. So in this reaction, exhibit the greater tolerance. In this, we are having some 11 derivatives. Among the substitute, we got the maximum yield, the 3H. We are getting around 90. So after that, however, we got the maximum yield 88% of yield in the 3I. That this side you can see here. Yeah, I think you are getting here. Yeah, these are the substrate code because we want to play different substrate electron withdrawing group or electron donating group. So what? So suppose just I'm telling generally, if you are using electron withdrawing group in this isotactic anhydride, maybe the reaction it will increase your yield, or it may be decrease your yield. Or suppose if you are changing in phenylacetylene side, same thing it will play. So after that, we want to change what is that? Changing intramolecular competitive reaction. So what does that mean? You can see here, I am taking this compound, isotactic anhydride, and you can see this phenylacetylene. It is one contained, as earlier I said, it's electron donating group and electron withdrawing group. This both in the same reaction vessel, the same optimum reaction condition. So which product is forming more yield? So what are the parameter, the driving force of the reaction? You can see here, if electron donating group present in the phenylacetylene, it forming the yield of the reaction is more. Is 1.8% is forming compared with the electron withdrawing group. Same like that, we can play here. See here, is aliphatic and aromatic. We are taken in the same reaction condition with the optimum reaction, the reaction condition. So which one is reacting fast, or aliphatic or aromatic? You can see aromatic that CH activation is working. I am getting the better result. So we have to check that the product, if you are using aromatic or aliphatic, what will happen the product? The product, which one is going, that reaction is a faster, the CH activation. So what are the major facts? If you see, already explained that CH activation, mainly the CH group, it is a thermally stable, is a molecule, means that bond. So thermally stable molecule, that bond is a breaking is very difficult. So how we are doing that CH activation? If you are using that catalysis, it will form the intermediate. It will that metal catalyst. It is weaken your CH bond. It will coordinate with your metal. Automatically, it will cleave that thermally unstable that bond. This is the purpose we are using that transition metal catalyst. I'm going to explain, but I think, yeah, the time is there. If you are using any traditional method, you can see, suppose I am telling the CH activation, simple reaction we can say here, H2. OET. This is active methylene group. We all are know that. This is also you are having CH, right? Because this CH is more acidic proton. So if you are using simple base, you can cleave this CH bond. This is traditional catalysis or reaction mechanism. Suppose if you are taking benzene, you are having CH, right? Or methane, CH3H. This is also your CH bond. This is non-acidic proton because this is thermally stable to proton. It is not possible to cleave the CH bond. So that purpose we are using this transition metal catalyst, means CH activation. What I'm explaining here, that thermally stable to the CH bond. What you are using the transition metal, it will weaken that CH bond. Then it will cleave. So it will form the desired product. This is the mechanism. So already explained. So this is the two major parties there. One is oxidative addition, reductive elimination. So firstly, you can see this is the first step process. The oxidation is coordinated with this ruthenium. It will form it's a rhodium complex, right? Rhodium cyclic intermediate A. Yeah, just a minute. This PPT. This is forming so rhodium complex. This is the intermediate A. Then after that, the sequential protonation and the cyclization provide the 6-membered rhodocyclic intermediate B. You can see here, you see, it will take protonation. You can see here. Oh, sorry. Our rhodium first is coordinate with this oxidation. It will form this rhodocyclic intermediate. You can see after that, so protonation is happening here. So you can see you get here NH2. It will form is a rhodocyclic 6-membered. You can see here, this is the 6-membered intermediate. After that, sequential addition of CO, you can see that, yeah, some problem. Yeah. So finally, our desired compound, this phenylacetylene, followed by the coordination with the phenylacetylene, this form the species, the intermediate 3. So finally, finally is insertion of aryl ring to the alkene afforded 7-membered intermediate. You can see here, after insertion of your phenyl ring, it will form 7-membered intermediate. You can see this is the 7-member here. This is the final version of the product. After that, reductive, already the reductive elimination it will take. So after that, you will form your desired product. This is 8 amino isocoumarine. This is the mechanism. I think if I want to explain, it will take more time because this is it's a vast field actually, the CH activation. Due to time constraint, I think I covered all the area. What is CH activation? So how it's working, and what are the advantage and disadvantage of traditional and conventional method. So after this is a mechanism. After forming all the product or substrate scope, just I want to go for the already explained. Just I'm going to check fluorescent property like live cell imaging studies. I'm taking some desired compound, some 8 compound. You can see this is my emission spectra. This is my AI data compound. This is aggregated induced emission. Because I'm not going to the depth because it will take some time. So just I want to show what is the CH activation, what are the application. So many applications that I am chosen here for cancer cell imaging studies. This is my one of the application, and we are doing different kind of application. One is photoluminescent studies or fluorescent property studies, and different kind of application already explained, medicinal or agrochemical or intermediates. I think I covered a little bit in that CH activation portion. So what are the in my chapter that I want to conclude, right? In this, I developed that methodology for synthesis of 8 amino coumarin with weakly coordinating isotonic anhydride with our existing catalysis. And we also studied some gram scale. So if you want to go for any industrial application, we want to give some gram scale synthetic approach. Means I think I didn't show you here. Yeah, this is the one what I chosen. Gram scale I taken 1 gram using this optimum reaction condition. So I am getting around 1.5 gram of desired product of around 75% of yield. So we want to do gram scale synthesis for any kind of industrial application. So already that also I did for gram scale studies. And also as the compound showed the fluorescent property, we studied the optical behavior of the amino isochromate solution state as well as thin film state also. And also we published in some good journal like MCOM or like that. So thank you very much for giving me this opportunity. If you have any question, feel free. I am ready to clarify it. Yeah, thank you. Hello.
Thank you. Thank you, sir.
Yeah.
Thank you, sir, for your presentation. Dear participants, if you have any queries, you can unmute and you can ask your questions. Sir, I think you have almost gave us a clear knowledge on the CH activation, sir. It is very interesting to know that applications of cancer cell imaging, and also we have got a clear idea on the fluorescence properties also, sir. Thank you, sir. Thank you so much. We are very delightful to have you with us. Thank you for spending your valuable time with us today, sir. Thank you so much, sir. Yeah. Gratitude is the fairest blossom which springs from the soul. Now, I call upon Dr. Irdia Antonin Sophia, Assistant Professor of Chemistry, to propose the vote of thanks.
Good morning to all.
It's my pleasant privilege to propose the vote of thanks at the end of the day 3 session of this 1-week online faculty development program on exploring new frontiers in chemical and biological science under START College program sponsored by Department of Biotechnology, New Delhi, and organized by Marian Star Center, Department of Chemistry, St. Mary's College, Autonomous, Thoothukudi. First and foremost, I would like to thank the Lord Almighty for his benevolent presence and blessings showered on us. I owe my special thanks to the resource person of today's session, Dr. P. Yuvaraj, Senior Scientist, CSIR, Northeast Institute of Science and Technology Branch Laboratory, Impal, Manipur, India. Thank you, sir, for sharing your precious time with us and sharing your expertise regarding the synthesis of biologically active small molecules using sustainable CH activation approach and their medicinal applications. Thank you, sir. Your session was very informative. My deep sense of gratitude to our secretary, Reverend Sister Flora Mary, our principal, Dr. Sister A.S.J. Lucia Rose, and our vice principal, Dr. Sister Colande Teres, for their constant words of encouragement and support. I owe my heartfelt thanks to Dr. Arakke Genesius Alphonse, overall coordinator and member secretary of this DPT Star College scheme. My special thanks to Ms. S. Rani Jaimeri, Head Department of Chemistry, and all the faculty members of the Department of Chemistry for their spirited teamwork and the coherent execution of the program. My thanks to the technical team for their uninterrupted support. Finally, I would like to thank all the professors and the teaching fraternity gathered here online from various parts of Tamil Nadu and also from various states of India like Andhra Pradesh, Karnataka, Maharashtra, Telangana, Uttar Pradesh, and West Bengal. Once again, I thank one and all gathered here online, this online platform. Thank you.
Thank you, ma'am. Dear participants, kindly fill the feedback form which was posted in the chat box. We thank you all for your continuous support. We thank you all.
Yuvraj sir, thank you so much.
Thank you.
Thank you,
ma'am.
sir. Thank you, sir.
Thank you very much, ma'am. This is my pleasure.
Okay, sir. Thank you, sir.
Okay, ma'am. Yes.